Good evening, everyone. My name is Sarquah, and I'm going to give a brief introduction about my research lab. As scientists, as we are, our overarching goal is to contribute to the betterment of this world. And for us to do that, we have to build on our chemical intuition. And according to the scientific community, for us to build our chemical intuition is based on our observation coupled with our theoretical models. And from this graph, we can see that when it comes to very simple and small models, we do have a very good understanding of it. But when it comes to big and complex models, our understanding is quite limited. This particular statement holds true in various fields of discipline or disciplines of science, such as in biology, chemistry, and in physics. But here then comes the question, how then do we bridge this knowledge gap? It is of this view that my supervisor in the person of Professor Thomas Schultz came up with a novel spectroscopic technique known as CRAZY. CRAZY is an acronym for Correlated Rotational Alignment Spectroscopy. And with the CRAZY technique, what we do is we correlate several spectroscopic technique data sets into a single data set. That is, we currently correlate the data set of mass spectrometry, the data set of electronic spectroscopy, and the data set of rotational spectroscopy. CRAZY technique is a type of form-proof spectroscopic experiment where what we do is we first align the molecules by exciting them. And after exciting them, we create a rotational wave packet, of which we then probe this wave packet. The signals from this wave packet is then sent through a time of flight mass spectrometer, of which the ions are being counted on a mass MCP plate detector for us to be able to determine our mass spectrum. The time delay modulation during which this mass spectrum is being detected or these ions are counted is then fully transformed to obtain our rotational frequency spectrum. So here's results from our experiment. For instance, we have this figure here shows the mass spectrum of acetylacetone, where you can see various isotopes from this mass spectrum. This time delay modulation of this various fragment isotopes is then fully transformed to obtain the rotational frequency spectrum of which you obtain over here. Out of this frequency spectrum, we are able to assign, fit, and determine the rotational constant of each of these isotopes. Out of them, we are able to then use those rotational constant to determine each of this structure, their molecule, as in determine the molecule of the structure. Unlike other spectroscopic technique that we already know, or the traditional spectroscopic technique we already know, with this CRAZY technique, what we do is, one advantage is it's able to produce or generate more than several information, unlike the other traditional spectroscopic technique where at one experiment you just generate one specific information. But with this particular technique, we're able to generate several information from just one single experiment. Not just that we are just doing one experiment after the other. In just one single experiment, we're able to generate more than enough information that we need on a particular molecule. So I will just end there and acknowledge my lab members or the CRAZY researchers over here, my professor over here, myself, and our postdocs over here. Thank you very much.